Great. I think we can get started. Thank you all for joining us today at the Connect to Science seminar series. A couple of quick reminders before we get started. First, we'd just like to encourage you to visit the website that's listed at the bottom of Andreas's title slide to get updates on the Connect to Science series, including upcoming speakers and access to some of the previous talks. Also, during the course of the presentation, please post any questions that you may have to the Q&A section at the bottom of the screen instead of to the chat box. So I'm so pleased today to introduce Professor Andreas Strasser from the Walter and Eliza Hall Institute in Melbourne as today's speaker in the Connect to Science series. Andreas is one of the most innovative thinkers in the field of apoptosis research and has pioneered studies to explore the regulation of this critical cell death pathway in mammalian development, hematopoiesis, and of course, cancer therapy. His many discoveries in this area, as evidenced by over 350 peer-reviewed publications, have elegantly demonstrated the potential for treating cancers by targeting the apoptosis pathway and have led to the preclinical and clinical development of some of the first BH3 mimetics, including venetoclax. He is now leading major efforts to uncover mechanisms of resistance to these agents and also investigate the toxicities that arise and how they are to be avoided. Those are some of the things that he's going to be talking about today in his talk. I also want to mention that Andreas has been particularly supportive of the many graduates from his laboratory and nurtures their research and careers with untiring resolve. I've talked to many of them, and they all mention the same thing about the support that they get from Andreas. It's so wonderful to hear. We're so glad that you joined us today, Andreas, and we turn the floor over to you.
Thank you very much, Chris, and thank you very much, Claudia, to organize this meeting. And I'm really honored to have been invited for this talk. So today, on advice of Chris, I'll start off with some background from a very, very long time ago that got us into the field of programmed cell death and its implications for cancer. And I'll end up on a whole bunch of unpublished data. I hope you'll like my talk, and yes, I'll encourage you to ask as many questions as you wish. I do have to make the following two disclosure statements that are on my second introduction slide. And so I am an employee of WeHi, and our institute receives milestone payments and royalties from Genentech and AbbVie for the development of venetoclax. And also we do collaborate with the company Servier on the development of MCL1 specific BH3 mimetic drugs. So you all know this very famous diagram from Hanahan and Weinberg in their famous Cell Review, that evasion of cell death, or evasion of apoptosis maybe more specifically, is needed for the transformation of a cell from a normal state into a highly malignant state. So what actually brought them to include that as one of the attributes of malignant cells? So it all started with patients such as the man shown here. This is a man suffering from follicular lymphoma, one of the more commonest forms of lymphoma in humans. So the work from Tsushimoto, and then later on from Stan Korsmeyer and also from Michael Cleary, showed that almost all cases of follicular simple lymphoma are characterized by a chromosomal translocation called T1418, in which the gene that was then named BCL2 for B-cell lymphoma, gene number 2, became subjugated to the expression control of the immunoglobulin heavy chain enhancer. The function of this gene was unknown. It was speculated that its protein would make cells divide when they are not shouldn't be dividing. That turned out to be wrong, and the famous work from David Vo and Jerry Adams and Suzanne Corey in 1988, which is depicted graphically on the right side here, showed that what BCL2 does is it makes cells survive when normal cells that don't express BCL2 or don't overexpress it are dying. So on the left-hand side, you can see a cell line that is dependent on the growth factor interleukin-3. If it's grown in presence of the growth factor at the top, the cells are all alive, shown in green color. When the cells are deprived of their growth factor, shown at the bottom, they all die, shown by the red and yellow color. The same cell line overexpressing BCL2 at levels similar to what is seen in follicular lymphoma, they grow normally in the presence of growth factor, they don't grow any faster than the control cells, but difference appears when growth factor is removed, then the BCL2 overexpressing cells don't die. So this showed a number of important things. It said that cell survival is controlled separately from cell proliferation, and that inhibition of cell death can be a central step in tumor development. And that was then very soon after was confirmed in vivo. So these are experiments where transgenic mice were generated that express BCL2 in B cells only, similar to what happens in the patient B cells. And you can see at the bottom in blue that overexpression of BCL2 on its own is relatively poorly oncogenic. If you're a patient and you wait for a year and a half, then you get a lymphoma incidence of about 5%. That is higher than control mice. But the great discovery was that if you co-express in B cells only MYC, an oncogene that makes cells divide when they shouldn't be dividing, plus BCL2, you get a very, very aggressive and very rapid development of fatal lymphoma and leukemia. So the mice are all dead by about 45 days after birth. You can see in the middle here a mouse that has such a lymphoma. So the lymph nodes are massively enlarged and the spleen is massively enlarged, and the blood is full of leukemic cells, as you can see on the far right side. So this led to the conclusion that on its own, blocking apoptosis is relatively poorly oncogenic, and that inhibiting apoptosis promotes tumor development by keeping nascent neoplastic cells alive, thereby increasing their likelihood of acquiring additional cooperating oncogenic lesions. So this slide here shows a lot of work summarized, and that is the discovery of all the relatives of BCL2. So we have in mammals, we have 5 proteins that promote survival, including BCL2, BCLXL, MCL1, which are the ones I mostly talk about today. Then we have in blue the effectors of apoptosis, BAX, BAC, and probably BOC. That permeabilize the outer membrane of the mitochondria and make cells die. And at the bottom in green are the 8 initiators of apoptosis, where presumably we would argue PUMA, BIM, and BID, and maybe BMF are the most important ones, and NOXA possibly as well. So this is the pathway of apoptosis as we understand it today. Again, this summarizes work from many, many labs around the world, much of it from Boston. And essentially, there's 2 pathways leading to apoptosis. On the right-hand side, the so-called DEF receptor or extrinsic pathway that is initiated by ligands that bind to TNF receptor 1 or the DEF receptor FAS. I will not talk about that today, so I won't go into much more detail here. And on the left-hand side is what is variably called the intrinsic pathway, the BCL2 regulated pathway, the stress-induced pathway, or my favorite name of it is actually the BAX-BAC dependent pathway, because activation of BAX and BAC, the effectors of apoptosis, really hallmarks that pathway. This is the point of no return. So in healthy cells, BAX and BAC are restrained and kept in check by the pro-survival proteins like BCL2 or MCL1, and the pathway is activated when the BH3-only proteins, the initiators of apoptosis, are activated transcriptionally or post-transcriptionally, for example, by deprivation of growth factors. The BH3-only proteins then, with very high affinity, bind to the pro-survival proteins, neutralize them, and that liberates BAX and BAC to permeabilize the outer mitochondrial membrane. And anything that happens downstream of that, such as the activation of the caspases, is really mechanisms to clean up the dying cells and make them die in a non-inflammatory manner. BAX and BAC can also be activated directly by at least some of the BH3-only proteins like BIM and PUMA, for example. So the BH3 only proteins are really, really important in the context of the treatment of cancers with diverse anti-cancer agents, such as the ones that cause DNA damage. So they do activate the tumor suppressor p53, which is a direct transcriptional activator of PUMA and NOXA. But DNA damage, which is not shown in this slide, can also activate some of the BH3 only proteins in a p53 independent manner. Um, um, Steroids such as dexamethasone can activate PUMA and BIM in a manner that is still not really fully understood. Even inhibitors of oncogenic kinases such as the BCR-ABL inhibitor Gleevec or EGFR inhibitors activate the BH3 only proteins, again, in a manner that is not fully understood, and they activate mostly BIM. So with this in mind, the idea really arose that if small molecule mimics of BH3 only proteins could be generated by chemists, then these could be used for cancer therapy because they would directly activate the apoptosis machinery by blocking up the guardians of cell survival, the pro-survival proteins. This was really the rationale that led to the collaboration between our institute and initially Genentech and later AbbVie to generate BH3 mimetic drugs. And one of the attraction we thought was that, well, such drugs, because they act in the cell death machinery downstream of p53, they should bypass the blocks in cell death initiation that are imposed, for example, by mutational loss of p53 that is so frequent in tumors. So this is a very historic and really exciting slide. So this is the imaging of the first person in the world with chronic lymphocytic leukemia who has received venetoclax, the BCL2 inhibitor, in the clinic. So you can see at the start of treatment, he had very bulky disease. This person had been heavily pretreated with other medications before, and at that time of entry into the clinical trial, didn't respond to any of these therapies. And his tumor shrank markedly over the treatment of the first 12 weeks with this drug. And then again, a very historic and exciting slide. This is from the famous paper that summarized the clinical data of the trials of venetoclax in Melbourne from Andrew Roberts and his colleagues such as Marianne Anderson and John Seymour. And it shows that in the cohort of patients tested, they all, or almost all of them, responded in all of their parameters of CLL disease, such as blood lymphocyte counts, the nodal mass, and the bone marrow infiltrate. And this then rapidly led to the approval of venetoclax for the treatment of refractory CLL. So while this was going on in my lab, we are fairly good at making knockout and knocking mouse models. We asked two very fundamental questions that we kind of were surprised why they hadn't been addressed before. And that was, well, although we knew that overexpression of BCL2 or overexpression of any of its functional homologs like MCL1 and BCLXL can drive tumorigenesis, for example, together with the oncogene MYC, it wasn't clear whether pro-survival BCL2 family members expressed under their endogenous control are really needed for tumor initiation, and which one of them would be critical in which cancer. And the other question that is more important from a treatment point of perspective is, well, once you have a malignant cancer, are any of the pro-survival BCL2 family members needed for the sustained expansion of that tumor, or to turn the question around, which one of these pro-survival BCL2 proteins should be targeted by a specific BH3 mimetic to achieve some treatment outcomes. So we developed models to address both of these questions, and I'll just give you some examples of what we have done and are still doing. So one of the models that we like using is the EMU-MYC transgenic model that I already briefly mentioned a few slides ago. So in that model, the MYC oncogene is linked to the hemoglobin heavy chain enhancer. So the cells expressing abnormal levels of MYC are the B lymphoid cells. And early in the life of such mice, that MYC overexpression leads to the accumulation of B cells, but that's not a clonal disease, it's a polyclonal expansion. But inevitably, with a mean survival of about 110 days, a clonal pre-B or surface immunoglobulin positive B cell lymphoma arises in these animals. And a model mimics a number of types of lymphoma, but probably best Burkitt's lymphoma that carries MYC immunoglobulin gene translocations. We think the model is more widely of utility because MYC is abnormally overexpressed in about 70% of all human cancers, way beyond leukemias and lymphomas. So to address the question of which pro-survival protein is needed for a development of a MYC-driven lymphoma, it's not as easy of just crossing BCL2 knockout mice with the MYC transgenic mice because the BCL2 knockout mice, they die about 30 days after birth from polycystic kidney disease. But in the lymphoid or hematopoietic system, there's easy tricks to get around that. So we generate E14.5 embryos, and the fetal liver of mice at that age is a very rich source of hematopoietic stem and progenitor cells. So we harvest the fetal liver and transplant that into lethally irradiated congenic black 6 mice. So the recipient mice, their blood cell system will then be derived of the cells that we transplant in, so such as EMU-MYC BCL2 knockout or all of the control genotypes you would need for such an experiment. So we predicted that loss of BCL2 would prevent lymphoma development because BCL2 overexpression accelerates it. So we're quite surprised that this was not the case. So the BCL2 is entirely dispensable for the development of MYC-driven lymphoma. As you can see here, there's no difference in the incidence of the disease in this experiment or the latency of lymphoma development. A much bigger impact was seen when we removed from the hematopoietic system of mice that have the MYC transgene, the pro-survival protein BCL-XL. So you can see here in red that mice in which BCL-X is absent in the hematopoietic system, they don't develop any MYC-driven lymphomas, as you can see here. So BCL-X is required for lymphoma development, whereas BCL2 is not. Loss of one allele of BCL-X, which you can see in the dotted black line here, didn't have an impact on the incidence or the timing of lymphoma development in this experiment. So by far the biggest impact was actually seen with loss of MCL1. So with MCL1, we can't do the fetal liver transfer experiment with knockouts because MCL1 knockout mice all die prior to implantation, so on embryonic day 3.5. So the only experiment we really could do easily was to make the mice just heterozygous for MCL1, which is tolerated. MCL1 plus minus mice are largely normal. So EMU-MYC MCL1 plus minus mice are almost completely protected from MYC-driven lymphoma. You can see on the x-axis the timing of the how long we followed these mice up. So the median survival of EMU-MYC mice in this experiment was about 90 days, whereas in the EMU-MYC MCL1 plus minus cohort, it was 351 days. So this is a massive, massive difference. So we then wondered whether taking away from this system the initiators of apoptosis would bring back rapid lymphoma onset. And indeed, it did. This is a relatively busy slide, but I'll go through it. So the the black curve is mice that have the MYC transgene and B cells with the median latency of about 100 days. In red, the furthest right curve is when you take away one allele of MCL1, so you really diminish lymphoma development massively. If you now, on this background, take away one allele of BIM, the light blue curve, you increase lymphoma development and accelerate it substantially. But most amazingly, in dark blue, which is kind of hidden under the green curves, these are mice in the B cells. They are MYC transgenic, MCL1 heterozygous, and BIM knockout. They actually develop tumors even much more rapidly than the control EMU-MYC mice. So the conclusion of this is that stresses that are imposed by MYC-driven neoplastic transformation in B cells must somehow activate BIM, the pro-apoptotic BH3 only protein, and thus a major function of MCL1 in tumorigenesis in this setting is to keep pro-apoptotic BIM in check. So we've done many, many more experiments on determining which pro-survival protein is needed for neoplastic transformation in various models of tumorigenesis, mostly hematopoietic malignancy, but this is where I'll leave that part of my talk and come to the second question, which is which pro-survival BCL2 family member is needed for the sustained growth of which type of malignant tumor. So to address these questions, we had to generate new experimental tools, and the diagram of what we did is shown here. So we generated mice that had the pro-survival genes BCL2, BCLX, or MCL1 targeted with LOXP sites, either in a heterozygous or a homozygous state. We crossed them with cancer-prone models such as the EMU-MYC mice that I mentioned, and we also had the mice to be heterozygous for a transgene that is called ROSA-CRIAT2, which is essentially a transgene that is expressed in all cells of the body of mice, and it encodes a tamoxifen-inducible CRE recombinase estrogen receptor fusion protein. So this fusion protein is inactive. It does not have recombinase activity in cells or in mice that are not treated with tamoxifen. But when you treat with tamoxifen, the recombinase becomes active and it recombines and deletes the LOXP flanked genes. So in this setting, these mice have normal levels of BCLX or MCL1 as they are untreated, and they develop lymphoma at the expected time, so median latency of about 100 days. We then extract the lymphoma cells quickly. We infect them with what we call our imaging virus, which encodes luciferase and GFP, and then we transplant these cells rapidly into multiple Black 6 albino recipients that are fully immune competent, and we then image the mice over time to observe the development of the tumor. And then when we can see a good tumor burden, we treat with tamoxifen to remove the BCLX or MCL1 genes and then look what happens to these animals. So I mentioned to you before that BCLX is absolutely essential for the development of MYC-driven lymphoma. So we predicted that taking away from these lymphomas, when they're established BCLX, the tumors would just shrink and the mice would be cured. Now, that was absolutely not the case. So you can see here a group of 4 animals. They got exactly the same lymphoma, and one of them was treated with vehicle. The other 3 were given tamoxifen, 4 doses on days 7, 8, 9, and 10 after lymphoma transplantation. And at most, there was a slight delay in lymphoma expansion in these animals that didn't actually translate, as you can see on the right-hand side, in an enhancement of tumor-free survival of these animals. So that was very surprising. We did not predict that at all. A very different story came out when we treated mice that have tumors in which we can remove MCL1 with tamoxifen in the tumor cells. So again, the setup is the same. So these are 4 animals. They all have the same immune lymphoma in which MCL1 can delete it by activating the CRE-RT2 recombinase. You can see the mouse that got vehicle progressed very rapidly, and we had to sacrifice it for ethical reasons. But the 3 animals that were treated with tamoxifen to remove MCL1, they actually were cured of the disease. So we did, Gemma Kelly did this experiment with many, many different lymphomas of each of the genotypes. So the large N number is the numbers of independent lymphomas of genotype that she examined, and the small n number is the number of independent mice bearing such tumors that was examined. So this is a very large experiment. And when you exclude lymphomas in which the floxed alleles of MCL1 was not recombined, which occurred sometimes because there was selection for that, you can see that removal of both alleles of MCL1, 100% of the animals survive long term. But even more excitingly, just taking away 50% of the MCL1 protein by taking away one allele of MCL1, we have long-term survival of 95%, 94% of the animals. So this showed us that these MYC-driven tumors, they could not tolerate even loss of 50% of the levels of MCL1 that they express. So we wanted to extend that to human lymphomas and leukemias. So to be able to do that, Marco Herold in my department, he developed a very elegant inducible CRISPR gene deletion model. So in that model, we express Cas9 with a M-cherry reporter constitutively in cells, and we induce the guide RNA of interest in a tetracycline inducible manner, and that vector has a GFP as a marker expressed constitutively. So we can easily select for cells after infection that bear both the expression vector, the guide RNA expression vector, and the Cas9 expression vector. The model works extremely faithfully. So when we use that model in, for example, in human Burkitt lymphoma cells, which are MYC-driven tumors, in culture, you can see that there is a very nice correlation between the level of survival that we see in these cells and the indels, basically the non-homologous end-joining events that occur in these cells, as shown on the top right-hand here. And again, that correlates actually with the level of reduction of the MCL1 protein, as you can see in the Western blot at the bottom. So taking away MCL1 also kills the human MYC-driven lymphoma cells in culture. This system even works in vivo. So these are now NSG mice because they're human tumors that we transplant into mice. And then if we activate an MCL1 targeting guide RNA in the tumors as they're growing in mice, you can see on the left-hand side that the tumors shrink. Whereas if we inducibly express a control guide RNA, then the tumors do not shrink. They actually progress during the 5-day observation period shown on this slide here. So this summarizes a huge amount of work from many, many different labs, notably also from Brian Koss and Joe Opferman, which shows that a large number of diverse tumors driven by different oncogenes and tumors of different cellular origins when you remove MCL1 in vivo or in vitro, these tumors shrink. Examples are acute myeloid leukemias driven by diverse oncogenes. Joe Opferman showed BCR-ABL-driven pre-BALLs shrink. I showed you the data of the MYC-driven B lymphomas. T-cell acute lymphocytic leukemia driven by either mutations in Notch or mutations in P53 are MCL1 dependent. Many other hematological and even non-hematological tumors as well. I just showed some references here. So together with the observation from Meyerson and Berukim, which showed that about 10% of human cancers of diverse origin have MCL1 gene copy number amplifications that are somatically acquired, and showing that MCL1 is essential in tumors, and data I've shown you here. If you put that together, you would say that MCL1 is probably a dependency factor in diverse cancer cells and may therefore be a promising drug target. So at that time, we didn't have available an MCL1 inhibitor that was highly effective. So again, we used the genetic tools to explore whether the removal of MCL1 also in normal cells would actually be tolerable. So to explore that, we used mice that are shown here. So these are mice that have 1 or 2 floxed alleles of MCL1 and this ROSA-CRE-RT2 transgene that I mentioned. So it allows removal of MCL1, 1 or both alleles in all of the cells of a mouse by administration of tamoxifen when the mouse is an adult. And then we test for pathology. So when we do this, then if we remove 1 allele of MCL1, which is the orange line here, the mice are fine. They have no ill effect that we can measure or observe, and the mice survive long term. If we do remove both alleles of MCL1, however, then all of the mice succumb within about 3 days after the first dose of tamoxifen. This death is not a hematopoietic cell loss related death, because if we give these mice a wild type hematopoietic system by transplantation that we do 10 weeks earlier, which is the broken red line, then the mice in which we now remove MCL1, both alleles in all cells of the body except the blood cell system, they also die. We're actually, to our embarrassment, still not absolutely certain why the mice in which we take away both alleles of MCL1 die. We still need help from experts on this, but we see a variety of problems. So we see liver damage evidenced by a substantial increase in liver enzymes such as AST and ALT. The creatinine levels go up, the serum urea levels go up. So we have evidence of damage to the kidneys, to the liver. We also have evidence of substantial muscle damage. We don't see, at least early, we don't see any cardiac problems, which was seen when MCL1 was removed specifically in cardiomyocytes, but not any other cells of the body. So again, we'll have to do much more on this. Well, while this was done, we started to get involved in a really exciting collaboration with the French pharma company Servier, which were the first to generate a MCL1 inhibitor that was highly specific and highly effective. So this compound is called S63845, and a very closely related molecule is in clinical trials at the moment. So the binding affinity of this compound to MCL1 is very, very high, and its binding to BCL2 or BCLXL is negligible, as you can see at the bottom here. The top right shows the chemical structure of this compound, which is incredibly difficult to generate. The A1210477 compound was the previously best MCL1 inhibitor, so this drug has a much lower affinity. And what's not shown here, it has a very poor permeability in cells, and it also has very high serum binding liability. So that drug is not even very useful in vitro. It can only do anything in vitro if you culture your cells in serum-free medium, and in vivo it does absolutely nothing. In contrast, the MCL1 inhibitor that was produced by Servier, it does work extremely well in vivo. So this was cohorts of mice that were bearing MYC-driven lymphomas that are MCL1 dependent. And when the mice are treated with the MCL1 inhibitor, the red line, then about 70% of the mice survive long term. You can see a clearance of the bone marrow space of the lymphoma cells. The control mice that were given vehicle, they all succumb to their disease, which is the black line here. This drug kills cells through an on-target effect. So this shows one of many experiments that we did. This is in the human H929 multiple myeloma derived cell line. So inducible expression of guide RNAs targeting MCL1 kill these cells, as you see on the top left. This killing is on target because on the bottom left, you can see that if by CRISPR, we render these cells deficient for the apoptosis effectors BAX and BAC, then they are not killed by this compound at all. And a Western blot at the bottom right just shows that we managed effectively to make these cells BAX-BAC double deficient. The top right here shows a measure of cell death. This is a cleaved PARP assay comparing directly the MCL1 inhibitor S63845 and the previously best known compound A1210477. And you can see that in terms of efficacy, there is more than 1,000-fold difference. This slide here shows that a very large fraction of human multiple myeloma derived cell lines are highly sensitive to this MCL1 inhibitor. In culture, color-coded the lines as highly sensitive in red, intermediate sensitivity in blue, and in green, relatively refractory. And you can see that there's no correlation between the oncogenic lesions underlying these cell lines, their transformation, and whether they are sensitive or relatively resistant to the MCL1 inhibitor. In red, I indicated 2 cell lines that we took forward to testing in vivo. You can see the data here, the AML1 cell line and the H929 cell line. And with various administration schedules of the MCL1 inhibitor, you can see that inhibiting MCL1 in the tumor cells and also the normal cells of the mice bearing these tumors is tolerable and has an excellent therapeutic effect, as you can see here. There's also acute myeloid leukemia derived cell lines from humans that are sensitive to the MCL1 inhibitor, as you can see on this slide here. Again, I color-coded what we consider highly sensitive lines in red, and in blue, the intermediate sensitive lines. And one of them we took forward for testing in vivo in SCID mice, the MV411 line, and it does respond very potently to the MCL1 inhibitor treatment in vivo. RNA-seq analysis across a very large number of tumor-derived cell lines that were tested revealed that the best predictor of sensitivity versus resistance to the MCL1 inhibitor across the cell lines tested was the levels of BCL-XL mRNA. So high levels of BCL-XL mRNA predicted relatively poor responses, and low levels of BCL-XL mRNA predicted very potent responses to the MCL1 inhibitor. So this is interesting because sensitivity of cancer cell lines to BCL-XL or MCL1 inhibitor conversely correlates inversely with levels of MCL1. And this is, for example, bearing out in patients with CLL treated with the MCL1, the BCL2 inhibitor venetoclax. Now that one mechanism of poor explaining poor responses is high levels of MCL1. So essentially, the non-targeted pro-survival protein, when it is present in a malignant cell at high levels, can engender resistance. Across a number of solid cancer-derived cell lines, we did find that very, very few of them showed potent responses to the MCL1 inhibitor in vitro as a single agent. So you can see most of them are color-coded as green here. There were a few exceptions of high or intermediate responding lines across lung, breast, and melanoma cancers. However, if we combined the MCL1 inhibitor in culture, at least, with either a MEK inhibitor or a BRAF inhibitor, which is shown at the top and bottom respectively here, then we see very nice cooperativity or possibly synergy in tumor cell killing, as you can see in the red line, whereas the green line shows treatment with the MEK or BRAF inhibitor alone, and the blue line shows treatment with the MCL1 inhibitor on its own. So one issue that we noticed early on is that the MCL1 inhibitor from Servier has a considerably higher affinity for human MCL1 protein compared to mouse MCL1 protein. So the bifluorescence polarization, which is shown on the left side here, the difference is almost sixfold in affinity. And this, we believe, explains why the human MYC-driven lymphomas have a considerably lower IC50 than mouse MYC-driven lymphoma lines. So this brought about the consideration that we might be underestimating the efficacy of the MCL1 inhibitor by doing studies in mice, or also that we might actually underestimate more seriously the toxicity, the on-target toxicity to healthy tissues of this drug. So to develop a model that would overcome these limitations, and I'd like to note that the other since then published MCL1 inhibitors from Amgen and from AstraZeneca, they are actually the difference in affinity for human versus mouse MCL1 is even greater than for the Servier inhibitor. In the case of the Amgen inhibitor, it's more than a 200-fold difference, so that their drug actually doesn't even work on mouse cells. So we generated what we call our humanized MCL1 mice. So we manipulated the mouse MCL1 locus by retaining the 5' and 3' regions and replacing each of the 3 exons with the coding regions for human MCL1. So that actually worked. So the mice are actually viable. So they, the worry was that by putting human MCL1 instead of mouse MCL1 into the locus, we might get the embryonic lethality that the MCL1 knockout mice get at day 3 of embryogenesis, but that was not the case. The human MCL1 is expressed in the cells of the humanized MCL1 mice, as you can see on the Western blot on the left. And human MCL1 is slightly larger than mouse MCL1, so you can even see that as a shift in the migration of the protein. Cells from these mice are normally sensitive to drugs like etoposide or forbalester or ionomycin, as you can see at the top right here. But as we would predict, the cells are actually more sensitive to the MCL1 inhibitors. So the humanized MCL1 mouse-derived cells are shown in solid black, and the control normal mouse-derived cells are shown in open white here. We show data for both thymocytes and B cells. So having these mice available, we generated tumors on the humanized MCL1 background. So we crossed in transgenes that promote tumorigenesis, such as the immumic transgene. And on the right-hand side, you can see that as we would predict, the IC50 of lymphomas that are humanized for MCL1 on both alleles is considerably lower than the IC50 for control lymphomas arising in a normal wild-type mouse background, which is shown on black here. And then excitingly, when we do treatment experiments, so now we do the optimal experiment. So we take lymphomas that come from the humanized MCL1 mice, and we transplant them into humanized MCL1 mice, and then we treat the animals. So we can treat them with relatively low doses of the MCL1 inhibitors, such as 7.5 mg per kg, 4 daily doses. Then what we see is that we save about 40% of the mice. If we add into this treatment a low dose of cyclophosphamide labeled CP here, that on its own also gives about a 40% cure of the mice, we get an almost 100% cure of the mice. And that treatment does not cause any ill side effects to the tissues that we've examined. And there's kidney, liver, heart, and muscle are shown here, but we've examined many, many more tissues than that. So then a question arising is that with all cancer therapeutics that have ever been developed, resistance is known to occur. So what could be factors of resistance to MCL1 inhibitors? So to do that, we performed whole genome CRISPR-Cas9 screens. So we take cell lines such as immumic lymphoma cells, we install a constitutive expression of Cas9, then we transduce the cells with the whole genome, so-called USAR guide RNA library. Then we put the cells under pressure of the MCL1 inhibitor. When after 3 to 4 days, we allow the cells to recover. After 24 hours, we allow the cells to recover for 3 to 4 days in medium without the drug. We then collect the surviving cells and then do a genome sequence analysis of the surviving cells. So across 3 different independent lymphomas, the top resistance factors that we found are BAX and P53 every time. So the enriched guide RNAs are shown in red, depleted guide RNAs, and we haven't actually looked into those very well, are also found, and they're indicated in blue here. So here's the hit validation for BAX. So we generated lymphoma lines that are either deficient for BAX or BAC or both BAX and BAC, and 3 different lymphoma lines here. What you can see here is that the removal of BAX, which is in green here, gives somewhere between 10 to 30-fold resistance to the MCL1 inhibitor across all the 3 lines examined. A removal of BAC doesn't actually render the lines more resistant to this drug. And as I showed before, and as predicted, the removal of both critical effectors of apoptosis, BAX and BAC, which is in purple here, renders the lines almost 100% resistant to the MCL1 inhibitor. So loss of BAX does not actually cause in these lines a general resistance to cytotoxic agents. As you can see here across the 3 cell lines examined, we tested many more. It doesn't actually, taken away BAX alone, render these cells resistant to etoposide, as you can see at the bottom. You really, to get a profound resistance, you need to make the lines resistance in BAC and BAX. So that means that loss of BAX really causes a fairly specific resistance to MCL1 inhibitors and not across all cytotoxic anti-cancer agents. And that means that it probably explains the potent synergy that we saw between the DNA damaging drug cyclophosphamide and the MCL1 inhibitor in vivo that I showed you before. So what about hit validation of P53, which was a very, very prominent hit in our screen? So quite surprisingly, for us at least, removal of P53 from the lines actually increased the IC50 in the response to the MCL1 inhibitor by only 2-fold at most. So we wondered, is that actually really relevant? So the next experiment that Sarah and Gemma did is a cell competition experiment in vivo. So she mixed GFP labeled tumor cells that had been depleted of P53 with the same cell line not depleted of P53, the parental one not labeled with GFP, and then mixed the cells at ratios of 50-50 or 10-90, and then treated culture with the MCL1 inhibitor and then followed the response. And you can see in all cases, the lines that have lost P53 when treated alongside parental lines that have P53 with the MCL1 inhibitor get very rapidly, within about a day, enriched in culture. They go from a 20% to 100%. So that means that loss of P53 really gives these lines a considerable competitive advantage when they're treated with the MCL1 inhibitor. So very quickly, so the P53 actually came up as a resistance factor in the data that I showed you previously, where we showed that in vivo removal of one allele of MCL1 would be very effective in treating MYC-driven lymphoma. So the survival was not 100%, it was 94%. And in the 6% of cases where we had relapses, all of these tumors actually had mutations in P53, as you can see on this slide here. So whenever by Western blotting you find high level of P53 protein, that indicates a mutation in P53, and that's borne out here by the gene sequence analysis. So we wondered whether if you lose P53, whether lymphomas become resistant to complete MCL1 removal. So we generated lymphomas that are homozygous flocks for MCL1 and homozygous loss of P53. But as you can see in the broken black line here, these cells, when you remove genetically MCL1 both alleles, the tumors still die in vivo, and we get the more than almost 80% cure of the animals. So the cells can lose one allele of MCL1, but not both. So that would indicate that lymphomas with mutations in P53, you just need to treat them with maybe a higher intensity of MCL1 inhibitors. So to get an idea of what might be the underlying cause of how a loss of P53 renders cell resistance to MCL1 inhibitors, we did RNA-seq analysis and a whole genome CRISPR screen. So for that, we used the P53 activator Nutlin, which is an inhibitor of the negative regulator of P53 MDM2. So top hits in the RNA-seq analysis were interestingly genes encoding proteins that inhibit protein that are critical for protein translation, such as GRAB2. So that indicated to us that what p53 does, amongst many other things, that it actually increases the levels of GRAB2, which then shuts down protein translation. So that would then lead to a reduction in short-lived proteins, and MCL1 is a short-lived protein. In the CRISPR screen that we did, where we treated cells with Nutlin and then again put in the whole genome library, guide RNA library, top hits were p53, which we would predict, PUMA, which we would predict, the pro-apoptotic BH3 protein that is regulated by PUMA. But then again, we saw actually a lot of hits like TSC2 and NAR10 that are regulators of protein synthesis and mTOR signaling. So that predicted that p53 also transcriptionally activates inhibitors of mTOR signaling that again would drive down very rapidly the levels of MCL1 and other short-lived proteins. And it would also actually drive up BEAM expression. And that was borne out. So when you activate p53 by Nutlin treatment in lymphomas, shown with a plus here, the MCL1 protein levels drop very rapidly and PUMA comes up. And if you render the tumor cells defective for p53 by CRISPR, which is shown on the right hand here, there's absolutely no drop in MCL1 protein. So on the basis of all that, we would argue that p53 induces, when it's activated, killing of malignant cells through a variety of mechanisms. So direct upregulation of PUMA and NOXA is one way that would then inhibit all pro-survival proteins. But driving up inhibitors of mTOR signaling, such as GRAB10, TSC1, and TSC2, would also lead to a reduction in protein synthesis, and that would rapidly bring about a reduction in MCL1. So on this, I'll stop, and I would like to mention the people that I've mentioned throughout the talk who contributed and generated the work, the data that I showed, Gemma Kelly, Marco Herold, Marks Brennan, and Sarah Deep Stratton, and our collaborations with Servier. So thank you very much for your attention. I'm looking forward to your questions.
That was great, Andreas. Thank you for the awesome talk. It really went through the basic mechanisms of apoptosis regulation all the way through to the most exciting new developments in cancer treatment. So we do have some questions where we can go a little bit past 1 o'clock, so we'll try to get to these as we can. First one is, are there any long-term toxicities, for example, cardiovascular, to prolonged treatment with the MCL1 inhibitor S63845 in the humanized MCL1 mice?
So at doses that are sufficient to cure the mice of MYC-driven lymphoma, that we don't see cardiotoxicity. We have, of course, done maximal tolerated dose experiments in mice that do or don't bear tumors. And yes, we can kill these mice with the MCL1 inhibitor if we crank up the intensity of treatment. We don't think that cardiotoxicity is the primary cause of the disease and death of the treated animals. But again, to our embarrassment, I have to say that we're not absolutely certain what kills the mice. Our suggestion at the moment, what we think, is that it's actually intestinal toxicity, but much more work we have to do on that.
That's interesting. So some of the early data from the MCL1 inhibition trials have of course shown a signal of some cardiotoxicity. Do you think that that's then related to just other comorbidities that are occurring in patients that are being treated? Does that have to do with the age at which humans are being treated with these agents compared to what we're doing with mice? Do you have any other speculation about what could be accounting for the differences in toxicities?
Yeah, I think that is a likely explanation. We don't have any data on that. We have started to collaborate with a heart expert. What we're doing is she has several mouse models of cardiotoxicity, mild to intermediate and severe. And we're now treating, well, we're actually crossing her mice onto the humanized MCL1 background. And once we have these animals, we then treat them with low, intermediate, and high doses of the MCL1 inhibitor. And we'll see whether we then see much worse damage to the heart in these animals compared to what we've done in very young and very healthy black 6 mice that are humanized for MCL1 at the moment. So we predict that we will see increased cardiotoxicity.
Yeah, it would make sense. So I think there's a few people that are interested in the specificity for the loss of BAX as conferring resistance to MCL1 inhibition as opposed to BAC. Do you have a potential explanation for why that arises, and have you done any further studies on that?
We don't have any data to really convincingly answer that. We are very interested in it. We've been doing a lot of work on that. What I'd like to mention is we have seen this preferential dependence on BAX dependency for MCL1 inhibitor killing, not only in MYC lymphomas. We've seen it across many other tumor-derived cell lines that are MCL1 inhibitor sensitive. In some lines, it's the effect of just removal BAX is a little bit less dramatic than in the MYC-driven lymphomas, but always we find that removal of BAC alone doesn't do anything, and removal of BAX always gives us some level of resistance, sometimes maybe only 5-fold, but in other lines up to 50-fold. Yeah, we have to do much work on that space.
That's an interesting problem to investigate. So another question, have you looked to see if some tissues that express MCL1 higher, to see if some tissues express MCL1 at higher levels than others? If so, do you think that also affects the efficacy of MCL1 inhibitors for different types of cancers that arise from them?
So certainly in cancer cell lines, there is a fairly good correlation that the ones that have higher levels of MCL1 tend to be more sensitive to the MCL1 inhibitor than tumor-derived lines that are low for MCL1 protein. But as I showed, an even better predictor is actually the levels of BCL-XL. So lines that do express MCL1 either at medium or high levels that have low levels of BCL-XL are universally very sensitive to the MCL1 inhibitor, whereas lines that have high levels of both MCL1 and BCL-XL tend to be fairly resistant to MCL1 inhibitor monotherapy. But if you then dial in an additional drug that causes transcriptional or post-transcriptional upregulation of BH3-only proteins, such as cyclophosphamide or etoposide, then you can actually kill the cells that have high levels of MCL1 and BCL-XL. Or of course, if you treat them with an MCL1 inhibitor plus a BCL-XL inhibitor, you can also very effectively kill such cancer cell lines. The problem is that a combination of BCL-XL inhibitor plus MCL1 inhibitor is highly toxic in mice.
Yeah, we have other considerations for that. Also, Tom Look says, Andreas, great talk. Has the S63845 MCL1 inhibitor re-entered clinical trials in humans? It was held up for toxicity issues, at least for a while.
So I know that I'm not a clinician, so I'm probably not incredibly qualified to answer that, but I do know from my collaborator, Andrew Way, at the Alfred Hospital, that they are continuing to use it in trials of AML patients that they have started. And they are, I believe, hoping that they can restart and enroll additional new patients in it, but they certainly have been allowed to keep patients that they had started on the drug on that. from what I hear, there was much less of a toxicity signal with the Servier MCL1 inhibitor than with possibly some of the others. But yeah, please, yeah, I'd like to qualify that statement clearly that I'm not a clinician. And if you are interested, please email Andrew Way. I'm sure he'd be very happy to answer your question, Tom.
Thanks, Andreas. So we have some additional questions that were asked, but we're unfortunately out of time today. We'll email those to you. And Andreas, if you take a moment to respond to those individuals on their questions, that would be really fantastic. Thank you. And thanks again for accepting this invitation to talk. We loved hearing from you and the latest on the use of BH3 mimetics for targeting cancer. So thank you again for joining us.